So I'm really excited to introduce our speaker for today. When we started our new section of biomedical informatics about a year ago, Dr. Cunningham and I knew that our first recruit had to be somebody really on the international stage. We had to get a really big leader in biomedical informatics. And we were so lucky that Larry Hunter was interested in coming here to look at a position. Larry's been in the field for 30 plus years. He did his training at Yale. He spent the last almost 20 years at Colorado. He has been foundational in the development of the field of biomedical informatics, developing methods of knowledge extraction. He started some of the seminal conferences in biomedical informatics and has continued to be greatly involved in those meetings. He's been a great mentor and advisor already, even in his short time at UChicago. He has mentored and trained many students. We're so excited to have Larry on our faculty here. I would encourage many of you to reach out to Larry if you have questions about biomedical informatics or how to leverage knowledge extraction tools from large data sets. And he's going to give us a great, interesting talk today on knowledge-based biomedical data science. So over to you, Larry, and looking forward to hearing your talk and questions afterwards.
All right. Thank you very much, Sam. So coming to Chicago has been a bit of a shock to the system, including dressing for giving talks, which didn't realize I had to wear a hat and gloves, but so it goes. I want to talk to you today about a broad area of work. I'm going to give you a few examples to sort of give you the flavor of it. But I'm really only sampling things. And I can't get the slides to advance. That's not good. There we go. All right. So what I want to talk to you about is this. You're probably all very familiar with data science at this point, but the idea of knowledge-based data science is a slightly different animal. It's trying to bring semantics into data science work, that is the meaning of the words. And so knowledge-based data science is really a way of talking about computer programs that act as if they knew things. And we can get it at the end into a philosophical discussion about what it means to really know something versus act as if you know something. But I'm going to put that aside for this. And doing things, having computer programs that act as if they knew things, we can manifest this in various different ways. So the program can answer a question on exams or pass an exam. It can use existing biomedical knowledge to do things that researchers find useful, like perhaps ranking different potential hypotheses by the amount of support that they have. Or to interpret data in light of existing prior knowledge. And there's, of course, a very long history of that in Bayesian inference, which is really a whole method of interpreting data in light of prior knowledge. But that's a very specific method. And there are many others. And we often, in our daily work, when we're trying to understand the results of our own research, and contextualize them, we are interpreting data in light of our own prior knowledge. And so another way of looking at it is from a technical perspective rather than a functional perspective, not just what these programs can do, but how they do them. And the way they do that work is by having some sort of formal in the computer representation of knowledge, in this case, biomedical knowledge, and then can do operations on that formal representation of knowledge. And this is all, I wrote a review article now 7 years ago that lays this all out and sort of scopes the field, talks about what people do. Whoops. All right. But the way I'm going to narrow this down for this talk is to think about informatics tools that support you in writing the discussion section of a paper. So most bioinformatics tools are there to help you get from the output of some instrument, say a sequencer or EEG or something like that, take that raw data and turn it into results, something I could put in a table or in a graph that shows up in the results section of a paper. But existing computational tools outside of this area are not really all that good at helping researchers understand those results, to contextualize them, to see what they mean, how they fit in with other existing results and the like. And so there isn't very much support for, if you will, writing the discussion section of your paper using computer tools. And so what I'm going to talk about today are some computer tools that are there to help you write the discussion section of the paper, to interpret those results rather than to take raw data and turn them into results. And so I'll talk about two different approaches to generating mechanistic hypotheses that would account for the results that we see analyzing our data. And then I'm going to spend a little time towards the end talking about characterizations of open scientific questions and how computational characterizations of desired knowledge that we don't have yet could also be useful. And I will wind up with a few words about ChatGPT, which is, as an old-time AI guy who's been doing natural language computational work for 35 years, the last year and a half, 2 years has been a particularly interesting time. Everybody all of a sudden is interested in this work. Everybody has opinions about it. So I'm going to try and touch on it very briefly, but I'm happy to, in the question time, talk about it more. I want to start with something that I hope many of you are familiar with and has been used now for more than a decade, which is this idea of ontology term enrichment. So it takes the output of some experiment, often these are gene expression experiments, and then takes a list of genes, say differentially expressed genes in some condition, and then tries to find what that collection of genes, often hundreds of genes, have in common somehow. And it's a computational method for doing that. What we do is each one of those genes tends to, at this point, have a set of annotations about things that it participates in, processes it participates in, molecular functions it might have, subcellular locations it might be found in, that kind of annotation. Take those terms, often expressed in the form of ontology terms, gene ontology, very frequently, calculate some background distribution about how often you would be expected to see that many genes that were annotated to that term just at random. If I picked genes randomly, had 200 genes, and then calculate terms that appear more frequently than you would expect in that chance model. And you get a table, something like the one you see on your right, that says we saw response to chemical stimulus in this list of a couple hundred genes far more often. We saw it 14 of the 200 genes or whatever it was. It's far more often than you would expect to see by chance. There's a little p-value over there. And this is a common way that people try and get a sense of what a large collection of genes that are differentially expressed, say, are doing together. And this is fine. It's a little bit vague. And it's also a little hard to know what to do with this, right? So response to chemical stimulus, response to abiotic stimulus, are those different? Do they mean the same thing? Protein binding, well, yeah, I would expect to see protein binding in a lot of things, but it's helpful, but it doesn't quite get at as much as we would like. And so the results are vague, often obvious results. They are often not entirely relevant to the phenomena we're studying. It turns out to be the case that in an awful lot of these studies, you're going to see an inflammatory signal. And that may be as simple as the reason for that may have nothing to do with the experimental circumstance, but if you puncture somebody's vein to take some blood, you often get an inflammatory signal. And often people have inflammation going on for all kinds of reasons. You'll see that. So these differential analyses often produce sort of common things that you see all the time. If you do enough of these analyses, you sort of put them aside. But it really isn't getting at that need that people have to take that big gene list that I got and turn it into something I really want to talk about in my discussion section. So how are we going to do that? So what we'd really like are specific, meaningful hypotheses about what's going on in that big collection of data. Mechanistic accounts that identify what are the players, what are they doing, when are they doing it, where are they doing it, to what end? And I'd like to see evidence for those claims, not just some list of phenomena that I might want to pay attention to, but where have I seen this before? What literature makes me think this? What data? How did you infer this? This is the sort of thing, if computer programs could do this, it would be really useful. So one of the ways we get at trying to do that is using those formal computational representations of knowledge that I talked about a few minutes ago. And we need a lot of integrated, well-represented knowledge in order to be able to do anything like what I just described. So where are we going to get that, and how are computers going to be able to manipulate it? Well, the way that we have gotten to in the field to do this is building these things called knowledge graphs. And knowledge graphs are useful everywhere. There's a Google knowledge graph that you may be familiar with. It's, if you ask questions like, what year was George Washington born? You get an answer by Google going after its knowledge graph that has lots of relationships and entities in it like that. For biomedical applications, we build knowledge graphs that look a little bit like the one that I have up on the screen here that has phenomena in it like proteins and DNA and tissues and diseases and pathways. And for each one of those little blue boxes there, I want you to imagine there's an ontology underneath all of it. So there are hundreds of different cell types. The cell type ontology will tell you what they all are, give us names, standardized names for them. There are, in human beings, roughly 20,000 genes. They will all have DNA. They will all have RNA forms, messenger RNA forms, translated into particular proteins. So under each one of those boxes, there will be 20,000 different human, in this case, examples of those things. And they're all linked up using these kind of complex networks. And so we can use all of that formally represented knowledge to get at some of those things that I described a few minutes ago. Here's an example of how that works. So I don't expect you to read all the details in the boxes. It's not important. The only important bit is the boxes on the far left and the far right are both drugs. And what connects those drugs in the middle, looking for potential interactions between those drugs, mechanisms of a synergy. So what we found is these two drugs have a negative interaction. You get adverse events when you take both of them. We're trying to figure out why. And so what we can do is find paths between those two drugs that link a protein and a process to each drug. And so the little yellow box on the far left is the drug. The boxes that are near it are proteins. The sort of mass in the middle are processes. And then as you go further to the right, there's more proteins and then the other drug. And what we're seeing is these two drugs target proteins that have a shared process, and that shared process is a hypothesis about what the mechanism is the interaction between these two drugs that led to the adverse event. So we can look through these big graphs to try and find hypotheses about mechanisms. This is a very simple version of that, even though the graphs are large and it's a pain to go through them and all that sort of thing. But this is a simple task because there aren't that many paths from one drug to another that involve protein and a process. So it makes it possible to find them all. But finding long paths in general is really hard because there are so many of them. When you have a big graph that's all connected like that, there are very many different possible paths through it. And furthermore, most of those paths are not good mechanistic hypotheses. Okay, so the fact that we could take those two drugs, they both have a target that's a protein. Both of those proteins are protein. So they both have a link that goes to the protein node. And so knowing that the targets of these two drugs are both proteins is a path through the graph, but it's not a mechanistic hypothesis. So we have to develop what we call semantic search approaches. And I'm going to show you a few of those. And these semantic search approaches, there are various ways to try and develop them, but mostly what we do are we develop templates or meta paths, common paths, things like I showed you with the drug. The drug has a target. That protein participates in a variety of processes. If you find some other drug that has another target, but that the other target also participates in those processes, that would be a meta path. And then we could do that for all combinations of drugs or for drugs that we know have some adverse interaction. And so the example I'm showing you here is from a grad student in my lab, Brooke Santangelo's PhD work, and she's working in microbiome analysis. So she's interested in mechanisms by which changes in the biota in your gut can, she's particularly interested in gut-brain axis, but could have an effect in your brain. And so what you see here are a couple of paths from a microbe to anxiety disorder that are effectively hypothesizing how this particular microbe could play a role in an anxiety disorder through a protein in the bacterium that creates a metabolite that can be absorbed and so forth. That's what those mechanistic hypotheses are. I'm going to show you an older example from a grad student named Ignacio Tripodi, trying to do toxicology doing this, trying to come up with mechanistic hypotheses for why certain compounds are toxic. And this is important because in the last decade or so, these AI techniques, machine learning techniques, have gotten very good at producing predictions about whether some novel chemical entity is likely to be toxic or not. Unfortunately, the FDA has not used those in order to, or EPA, the regulators who worry about toxicity, have not been able to use those very accurate machine learning predictors because they really want to know what the mechanism of toxicity is before they're willing to take regulatory action. In order to set a safe level of some toxic compound, they really want to know, what is this compound doing? What am I trying to protect against? So a black box machine learning thing that says this compound is likely to be toxic, not so helpful. So Ignacio's work was to try and identify mechanisms of action based on gene expression. And so what he did is he took a toxicology textbook, this one in particular, and wrote formal knowledge representations, a lot like the one that I showed you using that knowledge graph, of each different mechanism of toxicity. And so this is a typical one. This is caspase mediated apoptosis. And so there are several steps here. There's some mitochondrial membrane permeability increase that releases cytochrome C, that activates a caspase, that causes apoptotic DNA fragmentation that leads to this intrinsic cell signaling pathway that ultimately leads in apoptosis. Okay, so that was curated out of that book. He wrote these down for a dozen different kinds of mechanisms of toxicity. And then he came up with a computational approach for identifying which one is likely from time series of gene expression assays on human cells that have been dosed with that compound. And so you dose the cells with the toxin, you identify differentially expressed genes at different time points. That's each one of these little vertical bars in the top there. Each row is a gene. And then we're going to compute a vector distance from the expression that we observed in the cell that has been dosed with the compound to each mechanism step. So the first mechanism step back here, positive regulation of mitochondrial membrane permeability. It's going to have in those annotations I showed you a whole bunch of proteins that are involved in that. We're going to look to see if the proteins we're seeing in the gene expression assay are the same as the ones that are involved in that step of the process. And we're going to do that for each step of each process. He came up with a little out of sequence penalty. So you may have noticed in that process, there's an arrow from one step to the next. They unfold over time. We have time courses of gene expression over time as well, but it's not clear that they're not going to line up exactly. The time course of the biological phenomena will not be the same as the time course of the gene expression measurements we've taken. So we don't insist on exactly the same ordering, but there's a penalty that says, you know, the caspase activation ought to come first, and so forth. And so ultimately you could score each mechanism using this approach. And then once you've picked a mechanism, you can say, here are the genes that I observed for each one of those causal steps. This is why I think this is the mechanism that's here. And so this is an example output, Tylenol. And I'm only showing you one of the 10 steps in the toxicity pathway here. But you get the idea that here is a mechanism of action, shows you very specifically which proteins are involved and which processes are involved. And furthermore, can make suggestions to try and validate this particular hypothesis. So you could do knockouts that would interfere with this process. You could see what those would be. And you could also, it'll suggest variants that would affect the toxicity of this compound that have been observed in human populations. And so this is a way of trying to get computational help to interpret or provide hypotheses about mechanisms. And it's doing it by using these knowledge graphs, right? And now I'm going to show you a more complicated version of this, where Tiffany Callahan built a big knowledge graph that was about human disease. And sort of focused in on disease and its treatment, looking at drugs mostly, and trying to figure out how we could do this to understand actual patients, individual patients better. And so she was interested in pediatric rare disease. And it turns out trying to build the knowledge graph she needed to build to do this work was a really difficult task. And she ended up building this ecosystem to build different knowledge graphs. Why is that? Well, because there is no one best knowledge graph that captures everything we know about all of medicine. Even if we could build such a thing, it would be too big to be able to do practical computation on, right? We're trying to search for paths and find, you know, nodes that connect different aspects of that graph. And if the graph is very large, especially if it's full of things that aren't relevant to the task we're trying to solve right now, it makes searching that graph very hard, right? So we want to build knowledge graphs that are suited to the task that we're going to put them to. We wouldn't want to use getting on now millions of taxa in the graph that Brooke is using to try and understand microbiome effects on brains that has, lists of millions of different bacteria in it to do this work for trying to understand pediatric rare disease, because we're not going to pay attention to any of that stuff. So she built this ecosystem she calls Phenolator for taking in information from a wide variety of sources. So one of the things that these knowledge graphs do is integrate information from lots of different places. So we're going to get drug target information from drug bank. We're going to look at relationships between diseases and molecular processes from Go annotations and things like that. Then there's a lot of preparation work it takes to make them all compatible with each other, and then different ways you could imagine hooking them together, and other kinds of things you could do to sort of test the accuracy of the networks you build. So one of the formal approaches here is a logical one, and you can use logical methods to test that the network you assembled is actually logically consistent and complete. But that adds a lot of extra nodes and arcs to the network. So you might want to do that and then take them out, processes like that. That's what Phenolator does, and it allows us to produce these really useful knowledge graphs. And oh, by the way, you can see up in the top corner, there are links to the software. So this is all open source, all freely available. You're welcome to use it. Lots of people have used this. One of the tricks in getting these knowledge graphs to work on actual patient data is to try and basically translate between the way that patients are talked about in patient records and the way that science is talked about in these resources that we use to try and make them connect to each other. And the way we ended up doing that is trying to map from a controlled set of vocabularies for clinical information that's pretty widely used for sharing clinical data called OMOP, and try and map from OMOP into the open biomedical ontologies, which are the ones that we use to build those knowledge graphs. And so she did this heroic work, which I would love to see more used, called OMOP to OBO, where there's, I forget the exact number, something on the order of 100,000 mappings, all of which were manually verified by human experts. So this was an amazing piece of work. And extremely useful if you want to do this kind of stuff with clinical data. And I'm just going to show you just very briefly how we did it, some work with clinical data. So what we did was take particular visits. Let's see if I can get a pointer to work here. Can I do this? See the pointer? Yes. Oh, good. So these are visit records from a particular patient. And so these are all, coded. Here's a date, there's some diagnosis codes here, a drug, and a lab. And so we can take all of those and map each one of those into that knowledge graph. And then on that knowledge graph, we built something that's commonly used these days called a vector embedding that takes each node in that graph and turns it into a list of numbers. And that list of numbers sort of reflects where that node is in the graph and sort of will have similar numbers to similar nodes at similar places of the graph. So if there are multiple drugs that could be used to treat asthma, each one of the vector representations of those drugs will be fairly close to each other in vector space and very far away from a drug to treat something else and even further away from a disease or some tissue type or something like that. So these vector representations are useful because you can do math on them in all kinds of ways, and they also express this sort of similarity. Vectors that are close to each other in vector space represent similar concepts. So we're going to use that to create patient vectors. So what we're going to do is take either individual visits or just append a bunch of visits together, take these diagnosis codes, drugs, and lab values, and turn them all into these vectors of numbers, and then add them all up. And that will be a vector that represents that patient. Or we'll take the ones from a particular encounter, and that'll be a vector that represents that encounter with that patient. And it still has the same effect of similar patients will get similar vectors. So we'll be able to see similarities and differences between patients by doing vector arithmetic on them. All right. And furthermore, we could look at how patients change over different encounters. And one side effect that I absolutely loved, was totally unexpected about this, is our IRB back in Colorado decided that these numbers, just the numbers themselves, were not protected health information. And so we could take the vector representation of a patient and put it up on the internet and do all kinds of computation with it and other things more interesting than putting it on the internet. But it wasn't considered protected health information. Stunning to me, but it's a perfectly reasonable interpretation. You can't go backwards from the vector to the underlying things that created it. And even the underlying things that created it is just a list of diagnosis codes and drugs and lab values. You can't get from there to a patient anyway. So it's wonderful. We did all kinds of stuff with this, but I'm only going to show you one example. And this is a way of clustering patients. So each dot in these things is a patient. And you can see, I don't know if you can read these are kids with PKU, sickle cell disease, congenital hypothyroidism, and cystic fibrosis, color-coded. Some of them have more than one. And what we did was just to see, okay, can we separate patients with different diagnoses or with different natural histories or various other combinations of things? Can we use just these patient vectors, the ones that we created, to separate them out? And as a comparison, instead of mapping them into this big knowledge graph that has all this underlying molecular basis for, what protein the drug targets and what proteins are associated with that disease and all that underlying stuff, we could just make vectors based on the raw clinical values. And so what you see on the left are vectors based on the raw clinical values, not mapped into the knowledge graph. The ones you see on the right were mapped into the knowledge graph. You get much more interesting stuff if you include the underlying molecular mechanisms. So, for example, you can't see really that sort of gray blob in the middle on the left, on the lower one, mixes together basically all of these different kinds of patients. It doesn't do a very good job of separating them out. And the ones that have the molecular mechanisms do a much better job of separating out the patients. It also does things like find the two major kinds of sickle cell disease. Those get separated very nicely. If you have the molecular mechanisms, they don't get separated if you're just using the clinical data. So she did all kinds of interesting stuff with this. I recommend that you take a look at the paper, but in the interest of time, I'm going to move on from this and talk about a few other things. So what I'm really interested in doing these days, and when I just got funding, I mean, literally last week, from Chan Zuckerberg Institute to do, is to look at some of this work. And what we did here is take something that's been a long preoccupation of text miners in the biomedical world, building those knowledge graphs. And the way that text miners do build those knowledge graphs is to try and find factual assertions in the literature, this drug binds this protein, and then turn those into knowledge graphs like that. And so we took that process and sort of turned it on its head. And so instead of looking for information in the literature, facts like this drug binds this protein, we looked for questions in the literature. We wanted to know what were open research questions, controversies, unknown mechanisms, the sort of things when you read people's papers, you tend to see these statements of desired knowledge or questions in the beginnings of papers and at the ends of papers. The ones in the beginnings of the papers are the ones the author thinks they answered with the work that they did, which they may or may not have done, but at least lay out why I'm going to show you the results that I have. And then the ones at the end of the paper are, gee, these results are really interesting, and they led us to ask a whole bunch of new questions about why we saw what we saw and this, future directions for new research. Very common in pretty much all biomedical publications, you will find those. And so we decided it would be really interesting to go after representing those questions. What are the questions in the literature? And Mila Bogoslav, who did this work and who, by the way, won the American Medical Informatics Association Award for Best PhD Thesis last year for this work, national award. Got started in it because she was trying to figure out what to do for her PhD thesis. She wanted to know what were the important open questions. And she was interested for personal reasons in prenatal nutrition And so she wanted to know, what are the important open questions in prenatal nutrition? And she started reading things and she started making notes, and she got far enough along and showed me this stuff. I said, gee, this would be really great. We should write a computer program to do this. And we were off to the races. And it turns out to be really, the questions themselves, not the answers, but the questions themselves turn out to be really interesting to all kinds of people. They're interesting to researchers who are trying to figure out what to do. They're interesting to graduate students alike who are really trying to figure out what to do. But it also turned out to be interesting to funders, which is why CZI got interested in it. So CZI's motivation is, look, the research landscape is constantly changing. It's hard for us to keep on top of it. And in particular, CZI has taken on rare disease as an important thing. There are, depends on exactly how you want to count, but somewhere on the order of 10,000 different rare diseases in the world. Even CZI, even Zuckerberg money is not enough to put the sort of funding it would take to really get a deep mechanistic understanding of 10,000 different diseases. So they're trying to find synergies among these different research areas. And there have been a lot of ways that people have tried to find synergies in doing rare disease research before. You can try and find overlaps in phenotype. Why this work is focused because of Chan Zuckerberg's interest and other interest here in monogenic epilepsies. Those things, what they have in common is seizures. And so we can look at the commonalities in phenotype and say, okay, we'll look at seizure disease and see if we can get synergies in research there. Another way we could look is to try and look at underlying mechanisms or molecules that are involved. So this particular epilepsy is involved in a variant in this gene. What are other diseases that are involved that have variants in that gene? Can we find synergies there? Answer, yes. But we thought we'd look for questions. What are the open questions in trying to understand these diseases and see if we can find synergies by going not after phenotypes or mechanisms, but after questions. And so this is from Mela's work. She was interested in, as I said, prenatal nutrition, and she went and basically wrote this code to go over the prenatal nutrition literature. She was particularly interested in vitamin D. And find all the questions that she could find in that literature and then organize them, classified them, built a big collection of them, which is what this diagram represents. And it turns out a couple of them were really sort of surprising to the prenatal nutrition researchers who were doing vitamin D work that she was collaborating with. And those are the ones in orange. And so it turns out that just by collecting and analyzing and looking at the questions, even experts in the field find things that they think are interesting and useful. And so we thought we would try and scale this up. And sorry, I already told you most of what was on this slide. So this is the question about shared phenotypes or molecular mechanisms. But the issue here is we can do this at scale. We can look at the entire biomedical literature and pick out lots of questions and analyze them with the power of computation and hopefully use that to be able to find these interesting synergies, find cross-cutting questions that really could increase research efficiency. All right. So that's basically what I wanted to tell you about the Ignorum. On Sam's recommendation, I tried not to dive too deeply into the computational details of this work. So I am happy to do so. I have a million backup slides. I'd be glad to show you equations and code and all that sort of stuff that he encouraged me not to. But before I do that, I do want to talk for just a minute or so about ChatGPT and large language models, because I think that everybody is sort of casting around for what to do in the face of these things and what to think of them. And so I encourage you to remember what they are. They are models of language. They are not models of knowledge, right? So what I talked about in the beginning is how these we're going to get computational representations of knowledge, how this DNA is end up being coding for this protein. That's a kind of knowledge. LLMs don't do that. Okay, what LLMs are statistical models of how words follow each other or how letters follow each other. And we've been doing this for a long time, right? The small language models we started off with, we'd look at, okay, how would I predict the next letter that was coming in a text? And if you don't know anything about the text, the best prediction you could make about the next letter in that text is just the background frequencies of the letters that you're using in, whatever language you're in. If you're in English, good guess, if you know nothing, is the next letter you're going to see is an E, because it's the most common letter that's used in our language. Okay? And if you knew one letter that was before it, then you could make a better guess. So if I knew the letter that came before was a T, I would increase the probability that the next letter is an H, because that happens a lot in English. And I might decrease the probability that the next letter was P, because that doesn't happen very much. Okay? And so I can take bigger and bigger chunks of what came before and make better and better predictions about what's going to come next. And it turns out that using these techniques, if you take almost everything that's ever been written by any human being and look at the sequences that you see in there, you can make really good predictions about what's going to come next. But that's all they are. They are predictions about what's going to come next. And if what comes next is the answer to a question, that was what the input was, that's a good answer to the question, but only because of the statistics of the language that went into building that model, not because there's any underlying knowledge about the actual answer to that question in the world. And that can get us into trouble. So one of the things that happens is you get these hallucinations. You get the statistical model predicts the next thing that comes, and it's not true. And furthermore, these are really good predictions. One of the things that makes, I'm going to step back for a second and tell you a story about beauty. So what makes something beautiful? For a long time, there have been, for time immemorial, thousands of years, people have written about what is the essence of beauty. It turns out that when you let psychologists and other researchers loose on this question, they come up with answers that are somewhat surprising. And one of the things that is most beautiful, that human beings see as most beautiful, is an average. How is that possible, you think? Beauty is rare, not average. But very few people are actually average, right? An average is a statistical thing. So this work has been done largely in faces. If you ask people to rate the beauty of faces, the more average the face is, the higher the score is likely to be for its beauty. It's true for men and women. It's true, there's a million other things that you might think averageness is a proxy for. There have been great studies to try and look at all of those things. Nope, it's averageness. And the more faces you average together, the more beautiful they look. And people who are really beautiful have faces that are very close to these population averages. This is true for our language models too, okay? The language models are producing things that are statistically highly probable given that thing. So they sound great. So they convince us with their hallucinations that it's kind of like a really good bullshit artist, okay? It's the language is so beautiful that it sounds persuasive. Look carefully under the hood. When you'd use this stuff has been used in science a lot. Often you get fake citations in there. We call them hallucitations. So it looks like it's citing a journal article, but there is no such journal article. One of my favorite things I did when they first came out was ask it to tell it, hey, ChatGPT, tell me about Larry Hunter. Wrote this very nice bio that was mostly accurate, and it said that I had written this book that doesn't exist and I never written. It was a cool looking book. I thought maybe I should write it, but it wasn't true. So a lot of the work that we are doing these days to try and make these language models useful in situations like in medicine is to try and improve their factuality, do something that gets them closer to real answers and to reduce these hallucination problems. And one of the ways to do that is using established facts. There's a method, it's called retrieval augmented generation, or RAG, that's very popular these days, that kind of combines search with language models. So you go back, you search, try and find the real answer to your question, like Google it, and then take the documents that came back and put them into a language model to produce a nice, compact, easy to understand, fluent answer to your question. That works somewhat. It's better than just using the language models. And one of the interesting possibilities here is to try and build a hybrid of language models and knowledge graphs, where the knowledge that the language model produces a lovely, fluent output of is based in a knowledge graph, where we know the provenance and have criteria for the quality of the facts that are in those graphs. And so that's a really useful thing to do. I have a proposal under review at the moment at NIH to try and do knowledge graph retrieval augmented generation using language models and do some applications with it. All right, I think that's enough for now. I will stop and let you know that we're doing this ignorance base for monogenic epilepsies, which I would love to talk with anybody about. We have this great knowledge graph construction, search, and generation techniques that several students, Brooke is not done yet, her microbiome work is ongoing, are doing work around generating mechanistic hypotheses from them. And I didn't talk about it today, but I'm really interested in how it is that people and other creatures come to care about anything, and what in particular they come to care about. And I have a new project going on the origin and evolution of caring, which I'd also be happy to talk about at some time. My email address is there. I'm pretty easy to find, and I would love to develop new collaborations, find new students, and meet all of my new colleagues. And thank you very much for welcoming me to Chicago.
We have time for questions, both from online as well as here. I have one question to start with. The work you presented on the Ignorum relies on being able to mine all of the literature, and a lot of companies make a lot of money by siloing off literature. So how do you get around that issue of the literature being proprietary for a certain embargo time? Are you allowed to mine it and then publish it? And is that, I know it's not maybe it's not an interesting question, but I'm curious how you get around it.
That's a great question, and it's a very interesting question because it's with the advent of these large language models, it's become a much broader question. Scientists mostly want their work to be used, and scientific publishers are mostly okay with other scientists using that work. And funding agencies have made some efforts in recent years to make the literature more available. So between open access work, so a lot of the literature is increasingly open access, means you can get at it, and depositions like PubMed Central, where so for work that isn't open access, if it was funded by NIH, there's now a congressional requirement that you deposit a manuscript that has the essence of your publication in a public repository. And for people who are associated with an institution like the University of Chicago, the rights you have to look at journal articles via the library can be used to also do text mining on them. And so I have a paper from some years ago that I wrote with a librarian about how to navigate text mining licenses with publishers as a librarian, won a nice library prize. Lots of libraries are using it. For the most part, it's not that hard. If you're associated with an institution that has a library that has subscriptions to the things you're interested in, it's not that hard to do the text mining work. That doesn't solve the problem for everybody. And it became an interesting problem because there are a whole lot of people out there in the world who are very unhappy about having their text being scraped up into ChatGPT, or even more so, their art being scraped up into the image equivalents that also do prediction, same stuff, but make pictures. And so there is now a real conversation going on about what is owed to artists and other owners of copyrighted materials that are being used to train these kind of models. And that's going to get played out in the courts. There are lawsuits, millions of them at this point, going on about, who should get paid for what. A lot of the owners of lots of copyrighted materials, so publishers, but also photo archives and all kinds of others, have made deals with a lot of these AI companies and have sort of switched sides and are now, well, we own your writing now, so we're going to get paid for it, but you're not. And so there's, as usual in these situations where there's a lot of money sloshing around, there are battles over who's going to get paid and who's not. But for these purposes, for doing scientific text mining over biomedical journal articles, it's not that much of a problem. BioRxiv is a great source. You have to be a little careful about BioRxiv and the other open, these are preprint archives, if you're not familiar. A lot of them have been poisoned. People have put false stuff in them. And you have to take these very open things with a grain of salt and make sure you're getting what you think you're getting when you're operating over all of them. So there are challenges, but bioRxiv is great.
Question in the back.
In the back.
Say it loudly.
So I'm going to repeat the question for the people online. Why monogenic epilepsies? We already know what the gene is there, and why not go after something where the ignorance is sort of richer? And I have both a scientific and a pragmatic answer to your question. I'll start with a pragmatic one. The funder wanted me to. So the reason that CZI was interested in that is they've taken on rare disease writ large. They have a program called Rare as One, but the first tranche of funding, which went out more than a year ago, went to monogenic epilepsies. So there's already a community that has been gathered by this particular funding entity, largely around patient advocacy groups, but more richer than that. So it brings together the people I need to do this work. One of the things we're doing around CZI is instead of taking our own intuitions about what is an open question or statement of ignorance in the literature, we're going to actually go to researchers, clinicians, and affected people and their caregivers to ask them what they think the important open research questions are, so we have a better sense of what the real ones are, and then use those to drive the design and implementation of our computer models. So having that community that we have access to was a good reason to work in monogenic epilepsies. But let me take on the scientific question. Right. Well, so the scientific question is just knowing a gene and having a clinical description of a phenotype is a long ways from knowing how that mechanism works, and therefore a ways from being able to invent new treatments or improve the quality of the treatments that we're giving. And so there's a real interest in those communities to both find new treatments either through drug repurposing or new chemical entities, and to improve in various ways the quality of care and the quality of life that the affecteds have. So there's lots of work going on. There's plenty of research in those communities. One thing that's nice from our point of view about the monogenic epilepsies is there's about 100 of them. And so that's a good number for us to go after. If we were diving too deeply into just one, I would worry about how generalizable our results would be. And if we were trying to go after all of medicine, I worry about trying to, drain the ocean. So it's a good size focus area. It's 100 different diseases, but we would expect the questions to have some commonalities because of the nature of seizure disorders. So I think it's a good place for us to explore this. It is not the only place we want to explore it. The idea is once we get better at this, we unleash it really on all of medicine, but one step at a time.
The questions from under Daniel? Thanks for your presentation. You talked a lot about what about behavioral? Are any knowledge graphs being generated around predicting human behavior or state behavior? Sure.
You could build knowledge graphs about any place. If you can show me factual information, I can try and assemble it into a knowledge graph. One of the reasons that a lot of the work that has been done in this area has been in, let's call it molecular biology, is because there are so many knowledge resources that we can draw on to integrate into these graphs. So there are really good resources about genes and proteins and pathways and drugs, and it makes the task of assembling a good knowledge graph easier. So to the degree that the uses you want to put a knowledge graph to are make connections to that molecular data, then absolutely we could look at, drugs in anxiety or something like that and be able to do a lot quickly. For more complex behavioral phenomena, the existing ontologies and the existing knowledge sources, I would guess, are not as good. And so since less is known and some of what is known is in less formal sort of databasey kind of form, it would be harder to go after the sort of things that your talk therapist might pick up on in a session. Doesn't mean it's impossible, it's just harder.
Any questions online? Feel free to unmute. One more question before we close. It's a pretty big question. NLP's been around for a long time, and we all are used to using NLP tools for getting information out of free text. When you think about text extraction, what are the things where LLMs are going to shine over NLP, and what are the things where you would say just continue to use your NLP approaches?
Where NLP works already, I would say there's no need to change. I would say that's probably not a huge area, but existing techniques have been effective in some ways, and they're cheaper and better understood than large language models. Large language models have tremendous potential to improve almost all aspects of text mining. I've been dragged to that conclusion by my students who keep showing me things that I think we have perfectly good ways of doing, and then they come up with some clever prompting strategy and it works better. And I'm frustrated by that, but oh well, okay, it does. I don't see any reason to resist them. One thing that has been interesting, I think recently, is looking at the performance of small, more intensely trained language models. So these large language models are large, okay, tens of billions of parameters. That means they're computationally expensive to run. And we've put enormous amounts of effort into developing hardware that makes it go faster and seem like it's not so expensive, but it still is an awful lot of computation. And it might be the case that we could, for text mining purposes and NLP on clinical records, might want to use less computation than that. Or if we're going to try and run something over the entire biomedical literature, want to use less computation if we can. So small language models that are trained on particular narrow pieces of language have shown tremendous potential, really high performance. And so you get a little bit of the best of both worlds going that way.
So are we starting to see the commoditization of the development of these models, where you could basically take a non-highly computational person who take a corpus of literature and develop a model against that?
Yes, that's definitely coming. And one of the things that I'm for 30 plus years been a big open source, open access advocate. I don't think it's science if you do it in the dark. There's no such thing as secret science. But that's been a battle to fight. And there's been a really fascinating evolution of that battle with the advent of large language models. Because I don't know if you're familiar with this, but there was a bill introduced in California that basically would have banned open science, open source research on language models because they're worried about bad consequences and getting into bad actors' hands and things like that. That failed. And if you look at the open parameters, not quite the same as open source, but open parameter models that are out there, they perform within a fraction of a % of the closed models. So things like Llama, which comes from Facebook, but is an open parameter model. So we don't know what it was trained with, but we know what the parameters are, and you can just copy them. And you can do fine tuning and other training from there. Works very well. It's almost as good as ChatGPT 4.0. The ones where the training data is also open perform a little bit less well, but they're still awfully good. And so one of the things that I think is interesting about this is, okay, we had a battle about whether we should be allowed to continue to do open science around language models. I think we've won that battle. You never know. But the ability for anybody to do this and to do it from scratch, really with affordable hardware at this point. You can't do a really large model with affordable hardware. It's really within the reach of almost anybody. And I'm excited about that. And I also think it's going to be really interesting to see what happens to these, highly capitalized AI companies that have no moat because anybody can do it. So.
Right. Thanks for braving the cold outside and inside.
Thank you.